So I think we'll start the session for today. So I'll try to share my slide and yeah, please let me know whether the presentation is visible or not and whether my voice is clear.
So your voice is audible and the presentation is visible.
Yeah, great. Okay. So again, I welcome all of you to the NPTEL course on Bioinformatics Algorithms and Applications. This is instructed by Professor Michael Promiha from IIT Madras. And I'm Sunanta Samante, PhD scholar at IIT Indore. And for this week's tutorial session, we will cover the concepts that was covered by Professor Michael Promiha in his lecture series videos, as well as some of the concepts that he covered in previous slides. And we will also try to see some of the problems and how can you solve them, which are relevant to your assignments, as well as the particular exams that you will be having, right? I think there was a notification regarding the exam registration today. So if you guys haven't missed it, so I think it should be in your email or somewhere. So to take a look at that, because I think the exam registration is open and the date of exam was announced as April 19, 2026, right? So you'll get all of the details there. And for the exam also, again, you need to focus on the relevant concepts that Professor Promiha talked about, as well as the kind of questions that you are facing in the assignment right now. For example, in MCQ type questions or numeric answer type questions and so on, right? Okay. So can anybody tell me what Professor Promiha covered in this week's lecture? So we'll try to go there, and we'll also try to cover some of the topics that that we left in our earlier session as well. So anyone, what was the content of the lecture?
Sir, in this week's lectures, Professor Govind talked about, in first lecture, he talked about the Blossom 62 matrix and PAM matrix and how to calculate the score of PAM matrix. Then he talked about different dynamic programming methods for multiple sequence alignment. Then in this week we saw the blast tutorial also and the conservation score for the multiple sequence alignment.
Okay, I see. Yeah, so those are the contents of this week, right? Yeah, so we'll go there. So I guess let us start briefly by covering what we started in our earlier TA session, I would say, right? So that was about something called the dot matrix. So basically, in this series of visual representation of sequence alignment, so there are, first of all, the simple kind of methods like dot matrix, right? And then when you go from the just simple visualization aspect to calculating the scores, then you will also you will also be introduced to different type of scores. For example, the match scores, mismatch scores, and so on. And then based on those kind of scoring methods, there will be particular kind of alignment. So for example, if you want to do a global matching or global alignment, and if you want to do a local matching or local alignment, so for those different kind of alignment methods, there are different algorithms that are already been widely used, and those are based on those match scores, mismatch scores, and indel scores, and so on, right? Okay, so for the dot matrix, does anybody remember how did you construct the dot matrix earlier? So it is basically a simple sort of alignment visualization, which can also give us some important ideas about where we are seeing alignment, in which subsection we are seeing alignment, and so on, right? And it can also be used for just a broad comparison between multiple sequences if you're constructing dot matrix between different pairs, right? So which pair is more likely close to which sequence is more likely close to which other sequence, and so on and so forth, right? So anyone remember what dot matrix was like? Like how to construct?
Sir, Sir, as you said, it was a visual representation. So in y-axis and x-axis, we put a sequence, and then if we see a match, we put a dot into the matrix. And for visualization, if we see a diagonal, we can say that this sequence is aligned at this point. Also, we can create a window to minimize random noise. So if we create a window, for example, 5, so I will put a dot in the matrix when I have three matching pairs in a 5 window sequence. So I can put there to minimize random noise. It's a basic visualized representation of how two sequences are being aligned.
Exactly. Very good. Very good. Yeah. So it's basically on two axes, you will represent the sequences, right? Sequences for the first one and the sequence for the second one and so on, right? So for example, in this way one and then in that way another, right? And then whenever you see a match or matching characters between the two sets of sequences, then you will fill that particular junction point or intersection with dots, right? So that's why it's called the dot matrix. So you will start from one corner, let's say, and you will start to fill every possible junction point where a particular dot is possible, right? If there is possibility, we'll fill that with dot. Um, and then whenever you see, as you said, whenever you see diagonal rows of dots on the matrix, then that particular section will uh, tell you that this particular area is matching. This particular area, if you let's say superimpose, not superimpose, if you just try to see whichever part is corresponding to the diagonal from both of the sequences, you will get to see which particular regions are matching, right? And then whenever there is isolated kind of dots, those are most likely random matches and not any significant kind of alignment. And it is a good visual representation of the match between two different sequences, right? So briefly, as we talked about here, so for example, here on both of the axes, we have two sequences, right? And we will just go one by one. For example, this A is matching with in the first sequence, and it is matching with the second sequence as well, because both have A. So we put a dot in this particular junction. Then whenever you see A versus A, for example, here it's C versus C. It's again C versus C, G versus G, C versus C, C versus C, G versus G, and so on. So that's how we know that you have uh, made a dot matrix basically in this way, right? And as you said earlier, whenever we see a long stretch of diagonal, for example, here we can see these three particular dots here, yeah, present diagonally. So that means whatever corresponding to this particular diagonal on both of the axes. So in this case, it will be this one. In this case, it will be this one. You see that those characters are matching, right? So it's ACG versus ACG on both of the sequences that we're trying to match. And then let's say if you want to see like in character matching kind of way, so it will be if you align those, it will be something like this, right? So that's basically how you can try to align these sequences visually just by looking at a dot matrix, although it is not the most accurate way of aligning, because uh, you are just putting a dot, you are not counting for any scores and so on, right? And as said earlier, you can introduce something called the window size and stringency, which are basically, instead of matching one character by one character, you will match a particular set of windows, and based on how rigid you want your match to be, you will introduce a stringency score or stringency point. So for example, here in this case, we have ACG versus ACA. So we will compare these two windows, and since we can see that AC, the first two characters of the first subsequence and the first two characters of the second subsequence, they are matching. So with a stringency of 2, we can put a dot here, right? And then you will consider overlapping windows for all of the possible three sets of subsequences, and then we will go one by one. You will place all of the dots, and ultimately, if you see the final result here, so what you will see is that the number of dots that are present on this particular diagram is reduced by a lot, right? So in this case, it is kind of filtering out a lot of noise by reducing the number of dots in the plot. And this could be a good way if you do not have any pattern that you would like to see for a particular set of sequences, then you can play around with the window size and the stringency and if you will some window skip size, right, window step size where you will not consider all of the overlapping windows, so on and so forth, and then you could get possibly a good sort of result in some of your permutations and combinations in this particular parameters, right? That will allow you a clear visualization of the similarities that you believe there will be in the particular sets of sequences. Okay. So in this particular case, again, we see two sequences. One is the amino acid sequences for the 5 lambda CI, and another is the protein sequences, sorry, nucleic acid sequences for the same, right? In this case, again, you see it is particularly filled with dots. So these are on the x-axis there is this fudge lambda ci, on the y-axis this is fudge p22c2, vertical, horizontal and vertical one, and in this case we are comparing the similarity between the two, and clearly as you can see, we can see one particular diagonal here, right? The same thing with the DNA sequence as well, and in this case you will say that okay, these particular region will have some sort of matching between these two sequences, and this could be a sequence or a region of interest, functionally speaking, right? So that is how the dot matrix is constructed. So can anybody tell me, let's say we are having a same length of protein sequence versus protein sequence, as well as nucleic acid sequence or DNA sequence versus DNA sequence comparison, right? So let's say we have 1000 AA and we are comparing it with 1000 AA, right? Amino acids in one dot plot, and then let's say in another kind of comparison we have 1000 nucleic acid bases, and then in another one we have 1000 nucleic acid bases, DNA bases, right? Okay, so once in one set of comparison you are doing 1000 amino acid versus 1000 amino acid, doesn't matter what, just take it as a generic value, right? And then in another case we are comparing 1000 DNA versus 1000 DNA on x and y axis, right? So in which of these cases uh, there will be more number of noise or more number of dots in the dot plot?
Sir, in cases of nucleic acids, because there are only four nucleic acids, so four base pairs, so A, T, G, C. So there is a very much higher chances to get a match between them. However, in case of amino acids, we have 20 amino acids, so there is a larger window size here. So there you will not be getting a match
Much matches, yeah.
as frequent,
Yes,
as frequent as nucleic acid.
yeah, correctly, because you only have four nucleotides, right? A, T, G, C. So that means the chance that one one random position some X base will match against any of these, that is much higher, versus you have here 20 naturally occurring amino acids, right? So in this case, in a particular position, the match being the exact same, that chance is much lower, right? So that means that in the case of nucleic acid versus nucleic acids, if you are considering just same number of length, sequence length, so it will be much more prone to noise, right? So you might need to uh, basically introduce some sort of window size or stringency values and so on. But in any way, if you're comparing two different organisms, so in the case of nucleic acids, of course, your the length of the sequence will be much bigger, because three codons for one amino acid, three nucleic acid bases for one amino acid, right? That's how you transfer. So that's usually not a problem, but be mindful whenever you need to use some sort of window size stringency, you should do so. Okay. Now, okay, so there was one link that I put earlier. You can go there and you can visit the website where you can play around with just a visual representation of dot plot in their server that these researchers have given us freely. Okay, so that's one thing. Now, in this particular case, again, what you are seeing is that you are seeing sets of sequences. So this is, I believe, a DNA sequence. Yeah, human LDL receptor using a DNA slider, so that's fine. Now on the left hand side, of course, you see that it's a much cluttered plot. It's a lot of dots are present here. Then applying some sort of window and stringency, you get something like this, right? So that's good. That's good. Now this is basically a same sequence versus same sequence match. Okay. So on x-axis you have a sequence. On the y-axis you have the exact same sequence, and you are constructing a matrix using that, right? So that's how you get this sort of plot. So anybody can tell me what this main diagonal, why is it like a continuous diagonal here? This big diagonal that you see, why is it exactly continuous?
Because it's identical.
It's identical, exactly, because you are matching the same character versus the same character, right? So that's good. On the upper left corner, you see some sort of small cluster of diagonals forming, right, in this particular region. So what could this mean?
Sir, repeat.
Yeah, repeats, right? So uh, dot matrix, it can be used, of course, between different sequences to compare and contrast, right? Wherever there is a presence of some particularly similar stages of sequences, but also on the other hand, within the same sequence, if there are repeated motifs uh, in the length of the sequence, then you can also use dot matrix to identify those, right? So in this particular case, you can see uh, since it's the same LDL receptor sequence versus the same LDL receptor sequence, you will see that, for example, this particular region, it's present here also, it's present here also, here also, here also, here also, here also, and so on, right? So that means that there is a repeated pattern of subsequences that is occurring in this particular region. So it can be used to identify repeats in the sequence when you are comparing the same versus the same, right? So in the case, if you look at the note for the figure 3.6, it says that for figure A, of course, you had a window of 1, stringency of 1. So the original dot plot, nothing was changed. But on the B plot, you will see they applied a higher window size of 23 and a stringency value of 7, right? So that means that a dot is placed on the graph at a given position only if 7 out of 23 residues are the same. So that is the stringency and the window size respectively. And you have to, of course, mix and match different numbers, and you do different permutations and combinations. And then uh, you will see that in some sort of uh, point, at some sort of values of window size and stringency, you will get to see the clear pattern in this particular plot, right? So here it says that basis for listing the actual amino acid repeats themselves by one of the other method of sequential alignment. So that's how the dot plot basically works. Okay. Yeah. Okay. So now we'll also try to look into some of the questions that Professor Komiha introduced or talked about similarly, right? And then we'll go from there to the introduction to different type of scoring, like for example match scores, mismatch scores, and so on and so forth. And then also we'll look into different algorithms for the alignment. So, okay. So anybody tell me the answer of this question in the chat? What could be a globular protein? Just try to keep it in the chat. Just look into it. So I see some C answer in the chat. Others also try to answer, and we'll just look at it. So globular protein, by globular protein, you will see that they are basically your spherical or oval kind of shaped protein, whereas the other kind of other type of proteins, for example, the fibrous proteins, they're kind of having elongated structures, right? There are several. I see one hand that's there, by the way. So you can unmute or type the question. For globular proteins, you will also see that they mostly have like a hydrophilic exterior, right? So that means that they are soluble in water, whereas the other type of proteins, the fibrous proteins, those are usually insoluble in water, right? And they play a role in the structural part or the structural uh, sort of uh, function, right? Structural support kind of system, right? So the fibrous proteins you will see they mostly are present in extracellular matrices, whereas the globular protein you will see they have in the cytoplasm or your intercellular systems or intercellular kind of environments that are present, right? So the example of any sort of globular protein will be these will be your enzymes, your different transport proteins, hormones, and so on, right? So in this particular case, it will be insulin, right? The others are part of the fibrous proteins. The others are considered as fibrous proteins. Okay. So now in this particular case, again, just as we talked about earlier, we are now trying to move forward from just simple visualization to also introducing different scoring methods. So anybody can tell why do you need to introduce this type of scoring? Why not just align using dot plots? What's the
Sir, because if we compare, in proteins, we compare a basic amino acid with a charged amino acid, negatively charged amino acid, we can see that if we put a negative amino acid in position of a positive amino acid, it would change its property. But if there are two amino acids which are negative, if we interchange between them, or if we interchange between two positive amino acid or similar kind of amino acid, they wouldn't have a much functional change or the structural change in the protein sequence.
yes, yes,
Also, if we change a if we have a if we change for purine to pyrimidine in case of nucleotides, so the score should be higher in case of that. But if we change between two purines or two pyrimidines, it should be lower because they are the structurally they are the same.
yeah. So I'd say the primary point is that by introducing scoring, you are introducing a particular system for quantifying how good the alignment is or how bad the alignment is, right? Uh, and the scoring also introduces a lot of nuance, right? For example, earlier in the dot plot, you only put a dot wherever the match is present, or you don't put a dot wherever the match is not present, right? But in the case of protein sequences or DNA sequences and so on, just exact character by character match. Other than that, there could also be same types of matches. For example, one hydrophobic group could mutate to another hydrophobic group, then that likelihood will be higher versus if one hydrophobic amino acid that mutated to a hydrophilic amino acid, right? Similarly, as you said, the charged amino acid, for example, if a positively charged amino acid was to mutate to a negatively charged amino acid, that penalty will be higher versus the positively charged amino acid mutating to another positively charged amino acid, right? So this sort of nuance you can introduce only if you have a particular scoring system in at play, right? So that's why it's important for quantification as well as for uh, exact details or nuance of the system, right? So in this particular case, we are not going to have a exact in this particular question. You are not going to have an exact uh, particular scoring for different types of amino acid to amino acid or nucleotide to nucleotide mutation or having it same but here we will just do a character by character match and mismatch. And then what we also have here, so the match score is 2, the mismatch score is -1. And we have also something called the gap score, right? So anybody remember what the gap score represents? Or what is the gap in an alignment?
Insertion or deletion.
Yeah, insertion or deletion or indel, right? So that means that in at one particular sequence, something was present which in the other case was not present. However, this is what the optimum alignment you have, right? So in this particular case, for example, you see at this particular point, there is a gap present here, right? So usually gap scores are much more negative than the mismatch scores. For example, here you see in this particular, just the example scoring, you see the mismatch score is given as -1, whereas the gap score is given as -2. Okay. So why is that?
So nature doesn't prefer gaps. They
Yes.
prefer mutations, point mutations, but not gaps.
Yes, exactly. So insertions or deletions which result in gap, nature does not favor that. It can, however, in some cases favor point mutations, which will be considered in mismatch, right? Mismatch values. However, if there is a gap that is introduced in this particular set of sequence alignment, then we have to penalize that at a higher value because it's unfavorable in a general sense, right? So in this case, I see a lot of answers. I think D in the chat. So what you do is that you first write down all of the characters, and then you try to see wherever you have matches, wherever you have mismatches, wherever you have gap, right? So for example, this one is a match, this would be a mismatch, this here is a gap. So you have total of 2 matches, right? So that's 2 matches times 2, that will be 4. You have 3 mismatches, right? So it will be -3 in total. You have 1 gap, so that's 1 gap penalty. The net score in this case was 4 minus 3 minus 2, which was -1, right? So answer D in this case. So this type of questions, simple questions, you can also get in your exams. Okay. Now, as we talked about earlier, so introduction of gap is unfavored by nature, right? So what's even more unfavorable is that introduction of multiple gaps, right? So for example, in this case, you see in this particular sets of matches and mismatches, you have 2 types of gap penalties. One is called the origination penalty, another is called the length penalty. So the length penalty is very simple. How many number of gaps or indels you have, that many number of length penalties you will give. But origination penalty is particularly when a new gap at a new position is introduced. Okay. So for example, in this particular case, right, uh, you have 2 continuous characters that are absent here, right? So these are 2 length penalties or 2 gaps, right? But they're continuous. So that means at a new position, no gap was introduced except for the very first one when you started, right? So at a new position, only at this time the gap was introduced, right? After this T versus G, there was a gap at the place of C. But then at the next place, when you have the T, so the same gap kind of continued. You can think of it in that way. So no new gap was introduced in this case. So in this case, you will have kind of 2 length penalties as well as 1 origination penalty for the gap that was beginning at this particular position, the arrow position. However, for example, if you take a look into something like this, so it has, of course, 2 length penalties, but the gap is not continuous here. So that means at 2 different positions, gaps are introduced. So you will give it 2 origination penalties because again, if there is a gap introduced at a new position, it's not a continuous gap. So again, that will be much higher disfavor by nature. Okay. So that's why we will give it multiple origination penalties. So this particular question is trying to find out what would be the best alignment and what would be the net score in each of this kind of alignment. Okay. So you have to choose among the given alignment options. So could you please try to work it out and then we'll see in a bit. Okay. So try to find out calculation. So in the case of sequence alignment, we have to first find out the alignment score, and only by that, comparing those, we will see the best alignment that is possible for our sets of sequences. Right? Now, the first alignment in this case, so you just do the calculation by finding out how many matches are there, how many mismatches are there, how many origination penalties are there, how many length penalties are there. Okay. So in this case, max score is 1, mismatch is 2. For origin of cap, that is -2, and then for length of the cap, that is -1, right? So in this case, how do you do it? So you see total number of matches, of course. Okay, so for the first one, you have 3 A's on both that are matching, right? So in this case, you will see 3 times 1, 3 for the 3 matches. You'll have 3 mismatches, and interestingly here in this example, the mismatch scores are given as 0, so that's kind of neutral value, not having a penalty of negative scores. And then you have 2 origination penalties. Why? Because the gap was introduced, a new gap was introduced at 2 different positions. You see in those 2 positions, new gaps were introduced, right? So you have 2 origination penalties along with the 2 gaps. You have 2 length penalties, right? So the origination penalty is -4, and for the length penalty, you have -2. So the net score, that would be -3, right? Similarly, if you work out the third one, what you will see is that it has 5 matches, of course. So that means that 5 times 1 is 5. Then you have 1 mismatch, that's 0. You have 2 origination penalties as well as 2 length penalties, right? So 2 origination penalties would be 2 times -2 of -4. You will have 2 length penalties, that is -2. So you will get the net score of 1. So -3, -1, and then for the third option, you will have 5 matches, 1 mismatches, right? How many origination penalty in this case? Anyone?
-2.
Yeah, -2. So there is only 1 origination penalty because only 1 origin of cap is there. So that will be -2, and then 2 length penalties for 2 characters that are having gaps, right? So you will get the net score of +1. So the last one is -1. The first and second one are -3, -1. Yeah, so the last one is +1, right? So the ultimately the best alignment score is the third option, right? So technically this one will be the best alignment among the 3 if you go by the scoring system that is provided in the question. So the more positive or the less negative, the better the alignment here. Okay. Okay. So another quick concept that Professor Gomez also talked about. So these are related to the classifications of enzymes, right? So there is something called the enzyme number. Okay. So if you talk about this 3.2.1.2, can anybody tell which class of enzyme should this belong to? we're kind of mixing and matching different uh, problems. Okay, so I different types of questions. So I see in one comment you are talking about origination penalties and length penalties. Can you explain it? So basically, as we talked about, and as all of you also talked about in the earlier sets of questions, uh, so the penalties are basically the basically the if you want to find out the best set of alignment, then you have to go by which likely outcome of this alignment would be favorable by nature, right? Or which likely character to character match versus mismatch versus whatever gap is present there, that would be favorable by nature. Now, in the case of match, so the character at the particular position is matching with the character of the particular position from the other sequence also. So that's of course favorable, right? Because it's staying the same. So that is why you have a positive match score. For the mismatch cases, you have when the characters are not matching. So in some cases, you penalize that with negative values, or in some cases, it stays neutral. Now the gap, that is the cause of insertion or deletion, or representation of any insertion or deletion, as you can see in this particular example here. So in this particular alignment that they have constructed at these particular two arrow positions, there are gaps present, right? So gaps represent insertions or deletions or indels at short. And then since introduction of any gap is highly unfavorable by nature, so that's why when you try to compute the score for the alignment, you give it a negative value. So for any cases, if there is indel, you give it a negative value. Now, for each gap that is present, you are giving it a negative value, that is called the length penalty in this particular case. But if there is gap introduced at a new position, uh, so that is coming under this origination penalty. So of course, presence of gap itself is unfavorable, but presence of gap at a new position, that is even more unfavorable. So at whichever position you have a new gap introduced, so you will give it an origination gap penalty, basically the origin of a extra gap or an extra gap, right? So for example, if you compare the second or if you compare the first and the last, let's say, so for the last cases here, for the last alignment case, you will see that two gaps are present, of course, here and here, but those two are continuous gaps, right? So there will only be one origination penalty because the gap point origin was at this position, and two length penalties. However, for the first sequence alignment, if you consider it, so of course two gaps are present, but both of those two gaps have two different origins, right? So here one gap was introduced, and here another gap was introduced. So it will have two origination penalties. So this is even more unfavorable, this kind of introduction to new gaps, than a set of continuous gap in this case. So to again penalize that particular thing in the alignment, you are introducing the origination penalty as well, right? That's the basics here. Now here 3.2.1.2. So it is a hydrolase. Okay, so this belongs to a hydrolase. Now, what the particular type of classifications system for the enzymes are? So firstly, you will have the class 1. Okay, so the class number comes first, right? That's the class 1. You have oxidoreductases. The class 2s are transferases, right? 3 is hydrolase, 4 are lyases, 5 isomerases, and 6 ligases, right? So in this case, since it's starting with this 3, so that means it's a hydrolase enzyme, right? And the particular enzyme commission number that you have, it has 4 characters, right? So a particular uh, character that is given at the first position, so that will be the class. It will be the first number. So if the first number is 2, that means you have taken compare with this particular table here. So 2 was a transferase, right? So that means it will be a transferase type of enzyme, right? Then you will also have a subclass, which is the second number. So what type of transferase it is? It is a phosphotransferase. Okay. Then you will also have the third number, which represents what type of phosphotransferase it does. So that's a hydroxyl group as an acceptor. So that has a classification. Again, the numbering you have referred to. So that's one. And then what is the phosphoryl group acceptor? That will be the fourth number. So that is D-glucose in this case. So this is, of course, 1, right? So that's the overall classification for this particular in this particular system for the enzyme glucose phosphotransferase, right? Since this was introduced in Professor Gomia's lecture, I see that since this was introduced in Professor Gomia's lecture, then these type of questions or these types of classifications you might try to remember a bit, because there could be something related to it in the exams also. Okay. Now, in this particular reaction, which is given here, so it talks about the gas exchange, right, between tissues and alveoli and so on, right? So you see the dissolved CO2. Then you also see this HCO3 minus, which is part of this chloride shift process. And then on the right hand side, you see how the exchange is occurring in the alveoli side, right, on the alveoli part. So you know that for certain enzymes to function, they also require certain ions to be present, for example, right? In this case, in the case of, for example, kinases, you require Mg2 plus ATP complex, right? So that's again, the Mg2 plus is kind of a required ion there. So that has this particular functionality. In this particular case, you have another enzyme called carbonic anhydrase, or Ca, which catalyzes this reaction, which is CO2 plus H2O to one H plus proton plus HCO3 minus plus Cl, right? So this Cl again is taking part of this chloride shift as the HCO3 minus and Cl. So this particular reaction, anybody tell which ion should be required for that? So try to answer it in the chat and we'll just see in a bit. Okay. Yeah, I see one hand in the chat with. Yeah, please go ahead. Do we need to memorize subclass? You don't need to memorize subclass. Just try to memorize the class, I guess, because that is the kind of main thing here that Professor Pumiha introduced here. Subclasses, there could be a variety of them. So that will be very hard to remember. But the main classes, for example, lyases, transferases, ligases, and so on. So I think just take a note of particular those values. The first the first value in the EC, the enzyme composition number. Okay. So the particular enzyme, that is carbonic anhydrase, there needs to be an ion for it to function. These kind of ions are also referred to as cofactors for the enzymes, right? So again, I have given the reference from where I took all of the pictures or questions and so on. So those are also available at the shared Google Drive that I uploaded and shared with the NPTEL platform. So I think you have the link to access those, and I can continue to give any sort of necessary resources there. So you can also take a look at that. Also many other additional resources, for example, some coding related books and so on, bioinformatics, how you implement Python in it, just basic Pythons and so on. Those kinds of books are also kind of there. Some PDF values that I could find, PDF files that I could find. So you can try to look at it. Okay. So these types of memory-based questions can also come. Just the ones that Professor Kumhagan talks about in his lectures. So in this particular case, the cofactor for carbonic anhydrase would be zinc 2 plus ion, right? So they have their own sort of functionality because these ions are charged, so it could be having some sort of positional support. For example, in the case of kinases and Mg2 plus ATP complex, the Mg2 plus works as a positioning for the ATP. So that the phosphoryl transfer can occur. So in different ways, these ions can sort of play a role particularly. Okay, so that's the point here. Okay, so here, for example, some of the main ones that you have, uh, you might have studied it earlier. So for example, Cu2 plus copper 2 plus, that is required for cytochrome oxidase. Ap2 plus ap3 plus, these are again cytochrome oxidase, catalase, peroxidase, etc. K plus, that's for pyruvate kinase. Mg2 plus, as I talked about in different kinases, for example, hexokinase, glucose 6 phosphatase, pyruvate kinases, and so on. Mn2 plus for arginase, molybdenum is there for dinitrogenase, and so on and so forth, right? Zn2 plus is again here for carbonic anhydrase, which we talked about in this particular case. And then you have also some sort of uh, different functional groups or chemical groups that are related to this particular sets of enzymes. So you don't need to memorize or remember all of those. These are just for some quick examples of the concept. Okay, and you can take a look into the Leninger Principle of Biochemistry book or the Human Physiology book that I referred to earlier. So this is by Sherwood. Okay, so you can take a look at those for further concepts. Okay, now so if you compare, for example, the protein sequences, so there could be certain amino acids that are present at a higher rate, right? Or higher percentage of those sequences. Basically, the content of this particular amino acid in the particular sequences, you will see certain amino acids are more prominent or prevalent. You will see certain amino acids are also less prominent or less prevalent, right? So in this particular question, again, it's kind of a knowledge-based question. You would say. So it's asking for which amino acids or which two amino acids are the rarest occurring amino acid in protein sequence. So that means if you see the content of this particular amino acid, so they will be very low, right, as compared to the others. So any guess, you can give it in the chat. Try to look at it. Okay. Okay. So if you compare the contents of amino acid, you will quickly see that the CYS 16 or the tryptophan, these two are the rarely occurring or the rarest occurring amino acids in protein sequences, right? So they are present very less amount as compared to the other ones. Okay, that's a knowledge-based question there. Now again, you have kind of the same type of question, so just try to figure it out quickly. So what will be the alignment score for three mismatches, two mismatch, one gap between the two DNA sequences? Here you don't have the origination penalty and length penalty. It's the very simple one with only one type of gap score. So let's try to calculate it quickly and we'll see. Okay. Okay. So as you see, there are three matches. So that means that 3 times 5 would be 15 for the match score in total. Two mismatches. Again, we are keeping it neutral here. So 2 times 0 will be 0. And then one gap. The gap score is much higher here. So one gap would be -8. So 15 minus 8 would be 7, right? So the net alignment score in this case will be option C. So option C here, right? Okay. So that's the different type of alignment scores. Um, and now again, you are looking at a concept that is also discussed by Professor Romiha, which is called PAM matrix, right? So PAM matrix is again a matrix where you will try to find out uh, the set of mutation or transition scores between one particular amino acid to another amino acid, right? So anybody knows what's the full form of PAM? Anybody remember?
Point accepted mutation matrix.
Yes, point accepted mutation. That's called PAM. Okay. So it was first basically constructed by Margaret Dayhoff, again, who is a very highly regarded person, a pioneer in the field of bioinformatics, right? And what Margaret Dayhoff and her colleagues basically did was that they chose 71 groups of closely related protein sequences. Um, and not only just closely related sequences, they had a particular criteria. Okay. So the criteria was that greater than or equals to 85% of similarity if the sequences are having, then those set of homologous sequences they considered for construction of this particular matrix. Okay. Again, PAM stands for point accepted mutation, right? Uh, and now, it is expected that in closely related sequences, the mutations will not significantly change the common function. If there are a couple of mutations, point mutations here and there, they might still work in a similar way. If you see in the nature, of course, among different organisms. So, for example, the same protein, similar protein performing same types of functions will be present in different organisms, which will have very high similarity, sequence similarity, um, and their functional sort of parts are will mostly be conserved, kind of, right? So these kind of observed amino acid mutations that are already you are able to see in nature. So of course that means that those were accepted by natural selection, right? So that is considered the accepted part of the point accepted mutation here. And how you derive the PAM matrix? It's basically by a phylogenetic reconstruction using some algorithm called maximum parsimony, which is basically the less or the most simple explanation for the construction of the phylogenetic tree or alignment that would be correct. And you have so basically a set of closely related sequences which will demonstrate some sort of mutation frequencies, right? Now, one particular PAM unit, one single PAM unit is first used, of course, that is to construct something called the PAM1 matrix. Now, anybody can tell me what is a single PAM unit mean, or what is particularly PAM1 corresponding to? So it talks about mutation frequencies, right? Of course. So what will be the single PAM unit, or what particular value or parameter does it refer to?
Sir, it refers to a matrix which is formed by comparing two very closely related sequences,
Mhm.
and to get this scoring for distantly related sequences such as once for example 250, we use PAM 250 or PAM 1000, so we use it to compare with distantly related sequence.
Yeah,
So
that's
we multiply matrix by itself
yeah.
for 250 times in PAM 250, and for in PAM 2000 we multiply it by 1000.
Yeah, so as you said, the lower value of the PAM matrix that is used for the closely related sequence comparison, and wherever you have the distant kind of sequences. So if you want to compare those, you have to bring in a higher value of PAM 250. And as someone mentioned, I think a couple of people mentioned in the chat, so it says one mutation in 100 residues. Okay, so one PAM unit particularly, that is defined as 1% of amino acid positions have undergone changes. So that means out of 100 amino acid, if that is the sequence length. So at one position, the character will be changed for the amino acids, right? So that means there is a mutation going on there. Okay, so that's fine. And then if you want to derive the PAM matrix values for a higher order PAM matrix, then what you have to do, you have to go for a multiple step procedure. Okay, so that you have to do via matrix multiplication. Uh, but if you have, for example, certain amino acids changing in PAM1 alignment, let's say, or PAM1 scoring system, let's say. So firstly, of course, you have to calculate the relative mutability. So that is the number of mutational changes from a common ancestor for a particular amino acids, and you divide that by the total number of such residues in the particular alignment. So the total number of residues in the alignment. Okay. Uh, and then you go for a normalization process, and then you do a log-based transformation based on log base 10 in this particular case for the normalized mutability rate, and you divided by the frequency of the particular residue. So do this log kind of transformation, and then you have to do a log odds scoring computation. So it is rounded to the nearest whole number and then incorporated into the substitution matrix. Okay, so this reflects the likelihood of amino acid substitution. Or on the other hand, if the matrix, if it is same character versus same character, same amino acid versus same amino acid, then it would represent basically the amino acid staying the same, right? If you want to go for a higher order PAM uh, matrix value calculations, then that is done by matrix then that is basically extrapolated from matrix multiplication. If you kind of do a quick calculation, let's say short stage of sequences, then you give it some values, and then you do a matrix multiplication, you will see in all of the subsequent matrix multiplications, um, the mutation rate, or the value for the same versus same amino acid, that will continuously start to reduce, and the value for same versus different amino acid, that value will continuously start to increase. Okay, so then again you can sort of get a parallel comparison that, okay, if you are going for PAM 250, then in general that would make sense that that will be distantly related sequences, right? Kind of that sort of comparison, right? So relatively, of course, the value for same amino acid to same amino acid uh, intersection, that will get lowered, whereas the value of same versus different amino acid intersection, that will get higher in any subsequent matrix multiplication, right? Okay. Um, so of course, if you ask like what is the size of the PAM matrix, what 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 answer would that be? The size of a PAM matrix, because it's a matrix, right?
So 20 by 20?
Yeah. So you are comparing kind of all of the amino acids against all of the amino acids. So it will be 20 by 20 matrix. That is the dimension of the matrix. And then if you want to get some sort of analysis or inference out of any alignment scores, of course you have to understand the scoring system of the matrix, right? So can anybody tell what is the positive values versus the negative values indicate in a PAM matrix?
The positive values are the high scoring pair, so there would not be a such difference if we mutate between those two amino acid, whereas if the value is lower and it's negative, so it means that there would be a significant change in the function or the structure of the amino acid with respect to this mutation.
So is the positive value indicating that this sort of mutation is possible, or is the negative value indicating that this sort of mutation is possible in nature?
It's a positive value.
Yeah, positive value that will indicate that these mutations are occurring more frequently than anticipated, or the substitutions are occurring more frequently than anticipated among the evolutionary conserved replacements, right? On the other hand, the negative scores will be for substitutions that occur less frequently. So if you have a highly negative value for certain amino acid to amino acid, that case, that will indicate that these particular mutations or substitutions are not favored by nature from whatever you saw in the closely related sequences, right? So that's basically what PAM matrix is. And then as I talked about earlier, so you have to extrapolate from PAM one value the other sort of matrices. Okay. So the other matrices, as you go from the low to very high order of the PAM matrix, so you will get to compare more sort of divergent sequences. Uh, and then from PAM1 matrix, you have to do matrix multiplication so to derive those scores. So for example, if you want to calculate the scores for a PAM80, if you have a PAM1 matrix, so you just multiply the PAM1 matrix by itself 80 times in total. That will give you the PAM80 values. Okay. And as I said earlier, how it works is that you will see that for every consecutive multiplication that you do in the PAM1 matrix, right, you will see that the same amino acid to same amino acid rate that will again start to reduce slowly, and then the same to different amino acid values that will start to increase slowly. So there also you can see that okay, for the more divergent sets of matrices, we have to use the higher order of PAM matrix scores, right? Okay. And here is a quick table here. So if your PAM number is 0, so that indicates that the observed mutation rate is percentage wise is 0 and the sequence identity is 100%. If the PAM number is 1, then that means that mutation rate is 1%, sequence identity is 99%. If it is 30 PAM number, so that means the observed rate is observed mutation rate is 25%, sequence identity is 75%, so on and so forth. Uh, if you go very high value, for example, PAM 250, you will see that the observed mutation rate is 80%, sequence identity is 20% only. So that means that it's very highly divergent because it's not matching, right? There is a lot of mutations, 80% of mutations is present there. So that means for divergent sequences, you can get this coding from this higher order of PAM matrices. Okay. Yeah. So that was the PAM matrix. And in this particular case, this is the log odds matrix of the PAM 250 scoring system. Okay. So here it is saying the PAM 250 scoring system. Okay. Now here in this particular box or the diagram, you will see that they are saying that amino acids are again grouped into the chemistry of the side chains. For example, you have the sulfide hydryls, and then you have the small hydrophilic as well. Cysteine is the sulfhydryl, the S, the serine, threonine, proline, alanine, and glycine. These are small hydrophilic. Then NDQ, then acid amides, basic, small hydrophobic aromatics, and so on and so forth, right? And according to the PAM 250 scoring system, they have given the values for each amino acid to amino acid change or mutation rates, right? So by looking at this particular matrix, can you tell which two amino acids are the most resistant to mutation from this particular scoring system? So which two amino acids are the most resistant to any sort of mutations in this particular case?
Sir, cysteine and tryptophan?
Yeah. So if you look at the matrix carefully, then you will see that for cysteine, so that is this one, right? And for tryptophan, let's say you can take a look into this row here. So the first column for cysteine, the last row for tryptophan. For both of these cases, you will see that majority of the or most of the substitutions that are there, they are highly unfavorable, indicated by the negative values, right? So cysteine and tryptophan, those are the most resistant to mutation. If you also compare the value for the amino acids being the same, so for cysteine it is 12 and for tryptophan it is 17, right? So that means very high value for the amino acid staying the same in that position for the different stages of sequence, right? That means they are resistant to mutation. You can compare different amino acids again. For example, if I say, so is it likely or unlikely that a glutamic acid is being mutated to a leucine? So anybody tell that? Glutamic acid mutating to a leucine, is it likely or unlikely, or favorable or unfavorable?
Sir, it's unfavorable. Unfavorable.
Yeah. So again, you have to find out the junction point for the glutamic acid and leucine. So for example, here you have the E, glutamic acid, right? And here you have the L for leucine. Okay. If you see the junction point, it is -3, right? So that means that this particular change or substitution or mutation, that is unfavorable because it is having a negative values. There will be some again which is zero values. That means that's kind of neutral in a way. Again, if you try to see, for example, by just basically comparing the side chain groups and their what to say, their particular features or characteristics, you can also see that it is sort of giving the same type of scoring as with your common intuition. For example, if it's a positively charged amino acid, let's say if it is mutating to a negatively charged amino acid, of course that will be unfavorable because it's changing the side chain group's characteristics, right? In this case, charge. So if you see, for example, um, let's find out one pair. So this is glutamic acid. Let's say an arginine is being mutated to glutamic acid, then you will see that it's a -1 score, right? So that's unfavorable again. If you see the arginine is mutated to glycine, again that is again having a -3 score, so that is unfavorable again. In some cases, you will also find favorability, of course, at certain parts. For example, if it's a glutamic acid and if it's an aspartic acid, so it's changing to an aspartic acid, that is a 3 value, right? Positive 3, so that's favorable. Again, if you compare, these are both negatively charged amino acids, so that makes sense that there will not be that much change probably in their functionality. So that's again possible, right? So with this kind of PAM matrix, you can get this coding, of course. You can get the interpretation of this coding by looking at the table. Okay. So that's the PAM matrix. Okay. So the dimension of a PAM matrix is, as we talked about earlier, it's a 20 by
20 by
20. Yeah,
20.
yeah, exactly. It's a 20 by 20 matrix, right? So anybody can give answer to this question. So for evolutionary divergent sequences, what we need to use in the case of PAM matrix? Among all of these. So yeah, I see 250 or the C answer here. So for evolutionary divergent sequences, you need to use a higher order of PAM matrix. In this case, PAM 250 is the most commonly used matrix, right? Okay. Uh, so there are certain assumptions while constructing PAM matrix itself. So any idea what assumptions those are?
So every mutation is taken independently with each other.
Mhm.
So if I take a pair of amino acids, so it is independent of on a different pair of a mutation.
Yeah. Okay. Yeah. So yeah. So I think you were. So that works. It's also taking into account or not taking into account the fact that in a particular set or in a particular position, whichever amino acid was present before, uh, that could have also been mutated from a previously occurring amino acid, right? So in this case, it is considering the mutation process as a Markov process. So Markov process's definition is that at any particular time point, whatever mutation is occurring, for example, from alanine to threonine or something like that, so it only depends on the identity of the amino acid at that position and that on that particular time point or from for that sequence particularly. So if the sequence 1 is mutating to sequence 2 at a particular position, so the sequence 1 could have also been mutated from a previously occurring amino acid, right? And there could have been multiple sets of mutations that had to occur before the sequence 1 or a starting sequence ultimately arrived, right? So in the Markov process, it does not count the history of the previous amino acids in the sequences. It only takes into account that, okay, this particular mutation is occurring and it is only dependent at the correct time or sequence 1 when the mutation is starting as per our calculations. Okay, so that is one sort of assumption that the history of the previous amino acids in that particular position there that is not taken into account. And of course, as you said, as we looked into it, the matrix for the distantly related proteins that has to be extrapolated for from matrices using for short-term changes, used for short-term changes. So we saw that PAM1 was constructed first. Then for the higher order PAM matrices, we uh, went on multiplying the PAM1 matrix by itself several times, right? So that means for the divergent sequences where we use PAM250, so that's not a direct observable score that was seen, right? That was calculated. On the other hand, it's a in contrast, right? In contrast to that, it's an extrapolated sets of scores, right? So that means the direct observability is kind of could be lost here. So it could represent, it could probably not represent the accurate values in that sense for divergent sets of sequences. So that's again one kind of shortcoming, you could say. And then again, it considers that for each evolutionary interval, the frequency of change of amino acids to any other is the same as observed in the short term. So, for example, if you take a sort of evolutionary period, so like millions or billions of years, millions of years, right? So for that intervals, you will see whatever was calculated for the short term amino acid changes. So it assumes that for a large set of large span of time also, uh, that frequency will remain the same. Okay, so these are some kind of assumptions for constructing the PAM matrix. Uh, and yeah, so another one is here. It relies on the direct observations of residue substitutions from PAM1 derived from closely related sequences. Okay, so that was what PAM matrix was like, right? Uh, so there was another matrix that was constructed very similar to it, where in the case of PAM1, we took into account the lower PAM score for the more evolutionarily similar sequences. But here on the other hand, you will see a certain difference with the next matrix that we are going to discuss, which is the blossom matrix, right? Yeah. So PAM matrix, as you saw, it was directly calculated from PAM1 values for closely related sequence observations, right? However, there was a lack of statistics for more divergent sequences. So in the case of PAM, this was matrix extrapolation. However, for more divergent sequences, for calculating that from the observables, they calculated something called the blossom matrix. So that was introduced here. This is called the Bloch's amino acid substitution matrix, and it was based on direct observation of every possible amino acid substitution in multiple sequence alignment. So it's based on direct observation for every possible amino acid substitutions, and more than 2000 conserved amino acid patterns represented by blocks of less than 60 residues that was used to create this blossom matrix, right? Now, in this particular case, the matrix is not extrapolated, but they are the actual percentage identity of sequences used for the construction of matrix. For example, in the case of blossom 62, so the 62 value in this case, that will indicate that the average identity is 62%, okay, between the sequences. And if you talk about the numbering or the ordering of the numbers and compare it with PAM, you will find out that the blossom matrices, they are numbered in a reverse order as compared to PAM. So that means, as in the case of PAM, the lower numbered values that is used for the closely related sequences, the higher values are used for the divergent sequences. In this case, the higher values will be representing more closely related sequences, and the lower values will be representing divergent sequences. Of course, because the value of the blossom that indicates directly the average identity, right? For example, 62 is 62%. So if you have a blossom 80, so that means that is 80% sequence identity. If you have a blossom of a lower value, that means the identity score is lower. So again, you can see that they are ordered in reverse, right? The PAM and the blossom. Okay. In the case of also a log odd score calculation is necessary, but as compared with PAM, instead of a log 10 based, here they use a log base 2 in the algorithm. And then again, the positive and negative values, they again indicate basically similar sort of pattern. So positive values, they will indicate the substitutions occurring more frequently, and the negative values indicate that they will occur less frequently than anticipated. Okay. So this particular table that is showing again a blossom 62 amino acid substitution. And again, if you compare with PAM, you will see a very similar type of values here. For example, you will again see the cysteine and tryptophan. They are the more resistant amino acids to mutation, because more often than not they are staying the same, particularly in this particular position. If you compare again same amino acid to same amino acid, if you compare different amino acid to different amino acid, you will see that the scores are kind of similar with PAM, right? For example. Um, so that's the kind of the values that we'll get from the blossom 62, right? Okay. Very very similar thing. For example, here lysine versus aspartate, positive versus negative side chain, right? So that's a negative score. That's a penalty here, right? And then for the case of, for example, something like valine to alanine, that's neutral, so on and so forth, basically. Okay. So that's blossom 62 matrix here. Again, a very similar question that we also talked about earlier. So what could be the suitable representation for closely related sequences? Which option? Yeah, so I see A answer in the chat. We earlier talked about that the lower PAM number that is useful for closely related sequences, because that is directly kind of extrapolated at a lower value or at a lower rate of matrix multiplication from PAM1, whereas the higher value or the for the blossom matrix, for example, blossom 80, so that indicates the sort of similarity, right? Or in the other case, we talked about average identity, right? So the blossom 80 will mean that higher level of similarity to identity is present here, right? So in this particular case, question on answer number A should be the option for the closely related sequences. Okay, that's fine. Um, Now here it is talking about again which of the following E values will be the most significant for aligning between a pair of sequences. Okay, so it's again trying to introduce something called the E values. So can anybody tell me the answer in the chat? We'll look into it just in just a bit. So the concept of E values, that's directly kind of underpinning the sequence or significance of a sequence alignment, right? So what will be the E value for the most significant alignment? So the E value kind of relates to the significance, right? In this case, I see a lot of D answers. So the D for the D option, right? Of course, it's 10 to the power minus 2. That's very low value. But technically speaking, right, the most significant for alignment between a pair of sequences, that should have an E value of 0. So if you just talk very specifically about the between all of the values that are given here, because an e-value essentially that represents the probability that the alignments resulting from a database search are occurring or caused by random chance, right? So of course, an e-value of 0 is highly unlikely. That's very true. But from the given values, that would be the accurate answer here. Okay. Now, what you see basically, if we just construct a kind of plot. So, okay, let's see here. So let's say if you have kind of a simple plot here on the x and y axis, right? So let's say this is the local alignment score, and then let's say on the y axis you have the number of sequences. Now what you will see in this particular case, you might see something like this. Okay, so this what I'm kind of showing here. So this would be a very ideal representation or ideal kind of plot for a alignment, right, or local alignment here. Okay. Now, in this particular case, you have a threshold value that you have set particularly, right, or there could be default threshold value, and you are saying, okay, if we have alignment scores greater than this particular threshold value, then we can say that this alignment is the alignment is going on there, right? The sequences are aligning in this particular position or something, right? So below the threshold value, whatever peak you are seeing, that is unrelated sequences, right? Unrelated sequences, and over the threshold value, whatever you are seeing, that is the related sequences, right? So of course, this particular small peak that you are seeing, that would be the alignment for the related sequences, right? And in this particular case, what you see, there is a very clear distinction between the unrelated sequence and the related sequences peaks, right? Or the area that they are covering, right? Below the threshold and above the threshold. But in reality, in a lot of alignments, you will not get this sort of clean diagram or clean plots, right? What you might see in a actual sequence kind of alignments. So let's say here again, we have the same thing. Local alignment score. Then just say you have again the same thing, number of sequences, right? So what you might see is that you might see something like this. Okay. So let's say this one thing, and then let's say uh, that is another one. So if you have like a threshold value of this, if this is your threshold value, okay, and if this is your unrelated part, related, and if this is your related sort of plot, right? You will see in a lot of alignments, there will be an overlap between the unrelated sequences plot or the unrelated set of sequences alignment plot and the related sequences alignment plot, right? Now, this particular in this particular overlap, whatever area that I am shading here, so that is basically giving you the number of false positives. Okay, so the number of false positives is basically over the threshold how many alignments you are getting, um, that is unrelated sequence alignment, that is unrelated sequences, but again you are getting an alignment score of over your threshold, right? So that is the number of false positives and essentially the indicator of the e value in this particular case. If you have a lowered e value, that means that this overlapping area and this particular region, that will be much lower. Ideally, if you have an e value of 0, So you'll get something like this, the first diagram that I drew, in where you have no overlap. So that means that is clearly distinguishing between unrelated sets of sequences and the related sets of sequences for alignment, right? Again, for example, um, if you have, let's say, an e-value of, if you have something like some alignment score, and then the corresponding e-value, let's say you have 0.3, and then on the other hand, you have, let's say, 0.03, you have, let's say, 0.003, and so on and so forth, right? So, of course, uh, this one will be much less significant. This will also be very less significant. This will be again more significant than the other two, right? Because the lower the e-value there is, the most sort of accurate alignment value you will have. Okay, so that's the interpretation of this. Okay. Um, okay, so in this particular question again, Professor Gumi also talked about something called the conservation score, right? So the conservation score is basically at a particular position in a multiple sequence alignment, let's say, what's the uh, provenance of a amino acid staying the same at that position throughout the sets of sequences that we're comparing, right? And what's the propensity of that amino acid occurring at the same position? What's the propensity for it uh, changing also? You can interpret those by calculating something called the conservation score. Okay. So Professor Brome had talked about the unweighted and entropy based measure for the conservation score. So can anybody calculate this particular one and give us the answer in the chat? So for calculating the unweighted and entropy based measure for the conservation score, that's kind of using this formula. Okay, so it's the summation of Fa of I times ln of Fa of I. So the Fa of I will be the frequency of that amino acid at this particular position. Okay, so Fa of I will be the frequency. So, for example, just quick example, what conservation score basically is. So let's say you have dot something like A S T, right? Then you have again dot. Let's say T L S T, right? Then you have, let's say, S L S T. You can let's say you have A L S T, so on and so forth, so on and so forth, right? You could have A, S, T, so on and so forth, right? So in this particular case, at this first position, you will see, let's say this is like a set of alignment, right? In the first position, in this multiple sequence alignment, you see uh, the amino acid for the first sequence is alanine, then for the next one is threonine, then for the next one is serine, for the next one is alanine, for the next one is alanine again, and if you let's say see another one, for example, if you have something like this, you will see that mostly for the first position, the alanine amino acid is staying conserved among all of these sequences that are compared here, right? Alanine is also sometimes substituting to a threonine. It is also sometimes substituting to a serine amino acid residue. So if this particular position, let's say position 15, so in this particular position, what would be the propensity of the alanine staying the same versus again alanine changing to a threonine, alanine changing to a serine, so on and so forth. Okay, so I see one answer, multiple answers I think. Actually, one answer 0 here in the chat. So yeah, in this particular case, the answer will be 0 because of course, if the same amino acid is staying the same, so that means what would be the frequency of score for this particular amino acid. So if, let's say it's alanine, if it is staying the same, that would mean that, and on that particular position, uh, alanine is present for all of the sequence that you are taking into account here, right? So that means that the frequency would be, of course, in how many sequences you see alanine, of course, in all of the cases, divided by the total number of sequences. Again, that will again give you the total value, right? So in this case, it will cancel out and give you a value of 1, right? So basically, let's say you will see the number of alanine at that particular position, the number of alanines you see, something like that, and at that particular position, or the total number of sequences. So let's say capital N, right? So you divide that by capital N. So in this particular case, since this both of these will be same, so it will be 1. So that means the ln of Fa, that will also be 1, right? So that means that if the particular positions are occupied for the same amino acids, the ultimately conservation score you will get, that would be 0. Okay. So that's
Can you repeat again? I didn't understand.
so in this particular case, for the unweighted and entropy based method for the conservation score calculation, so it will be this particular formula you have to use. This SA value, that is talking about the frequency. Okay, so the frequency of the particular amino acids, either it is staying the same or it is changing. Okay, so for example, in this particular case, if we have let's say at this particular position, if you have certain amino acids, let's say, so how do you calculate the conservation score? So in this particular case, you will have to calculate the frequency of alanine times ln of the frequency of alanine plus the frequency of threonine versus times the ln of frequency of threonine, then the frequency of serine times the ln frequency of serine, so on and so forth. You will just go for all of the 20 amino acids here, and ultimately you do a summation. However, if in the case, for example, in this case, the fourth number of position, whatever the last amino acids that I have indicated, in all of the sequences, you see there is a threonine present, right? So that means that, of course, you have to calculate only for threonine, and then what you calculate is the frequency of threonine for that position, right? So the frequency of threonine, how you calculate that? So the total number of threonines for that position in the sequences that you are seeing, I mean the total number of sequence where you see a threonine in that position versus the number of sequences that you're calculating for. So in this case, both of these will be the same, right? So ultimately, it will give the Fa value as 1, right? So since Fa value is 1, the ln Fa part, so that will give you a value of 0, right? So 0 times whatever frequency, 0 times 1, that is 0 still, right? And since there is only threonine, you don't have to calculate for any other amino acids. So ultimately, in this case, the whole calculation will give you a value of 0. So that is
Okay, sir.
yeah, that's kind of the unweighted and entropy based measure, as Professor Komia also talked about. Okay. Now, if you talk about sequence alignment, of course, there are broadly two types of alignments, right? One is global alignment, which is again, if you try to find kind of a global match, so you just compare over all the different sequences. Very useful for phylogenetic tree, something like that, evolutionary relationships, right? But there is another type of algorithm for aligning, which is called local alignment. Now, any idea what the local alignment is useful for? Just try to give it in the chat, and then we'll present. Okay. So, whereas global alignment is very good for overall sequence matching, right? Similarity matching. The local alignment is the best for, since it's since the term is local, so that means it's best for finding out short stretches of subsequence where we are seeing some similarity, right? So that means it is useful for identifying similar subsequence. If you remember, in one of our earlier classes, we also talked about something called motif discovery, right? So motif discovery is basically, motifs are basically stretches of sequences that are conserved across the similar group of proteins, across the different organisms, so on and so forth, right? So they could have some sort of functional relevance broadly. So those sort of motifs we can discover by performing local alignments, where they try to maximize the alignment score for small stretches of subsequences instead of focusing on the overall alignment for the sets of proteins. Okay, so that's local alignment. There are of course algorithms for that we will discuss in some classes. and then let's say if you have a global alignment, the value for each element in the matrix that is assigned using which of these options. So is it insertion, deletion, zero? Is it match, mismatch, insertion, deletion? Is it deletion, match, mismatch, zero, or match, mismatch, insertion, deletion, and zero? So this is talking about a global alignment, okay, not the local alignment part. Yeah, so I see some B answer in the chat. So for global alignment, if you look into the algorithms, you will see there that of course the match mismatch score that is considered, indels, insertion, deletion, that is also considered, but in this particular case, zero is not there. Okay, so the zero part particularly comes in the local alignment algorithm, not for the global one. Okay, so for the global alignment, it is match, mismatch, insertion, deletion. We will also see what the zero basically indicates, or how it is kind of incorporated in a local alignment algorithm, uh, where we talk about the particular algorithm, that is the Smith-Waterman, right? Okay, so here again, some question related to the E values. So it is asking that when aligning a query sequences comprising 100 residues in a database with a total number of 10 to the power 12 residues, the P value for the ungapped high scoring pair in a region in a match of the database is 1 times 10 to the power minus 20. So what would be the E value? So try to answer it in the chat and then we'll look at it. Okay. Yeah. So here there is one particular formula which is stating that E value is equals to M times N times P. Okay. Now what is M here? M is the total number of residues in a database slash sum of the length of the sequence, the total length basically in the database. And then N is the number of residues in query, and then P is the probability that the HSP alignment or high scoring pair alignment is due to a random chance. Okay. So if you multiply in this formula M times N times P, you get the exact E value, which will be 10 to the power 12 times 100 times 10 to the power minus 20. So it should be 10 to the power minus 6. Okay. So in this case, the option should be option C. And again, in this case, you try to remember this particular formula, that if you have been given and a total number of residues in database, this total number of residues in query, as well as the probability of the alignment being random. So you can calculate the E value using this M times N times P formula. Okay. Okay. Uh, Now let us talk about the algorithm that is used for the global alignment process. So anybody can tell me the name of which algorithm that is considered or used for global alignment?
Sir, global. Yes. Needle Wunschman.
Yeah, Needleman-Wunsch algorithm. Needleman-Wunsch algorithm is the one used for global alignment. Okay. So what uh, does the algorithm look like? Okay. Now, again, as we talked about earlier, for all of these algorithms to work, you need to have some sort of match scores, mismatch scores, and some gap scores. Right. Now, the first thing that you will do is that you will start at the first cell, the upper left corner, with a value of 0. Okay. Uh, and in this particular case, so in all of the other alignments or visual representations, you saw that the diagonals indicated sort of a match, right? So in this case, again, similar thing will occur where the diagonals will talk about match or mismatch, whereas the horizontal kind of skips or jumps, that will be considered as gaps or indels. Okay. So firstly, for this coding, you have to write down the gap scores for all of this. Okay. So the first row and the first column, and the gap scores will accumulate. Okay. So if you have, for example, one gap, of course you have a gap penalty. If you have a gap again next to that, so that will again count as two gap penalties, right? So the gap scores or the indel scores, the penalty will accumulate overall. So how you do that is that first you go for this particular cell, the particular row, where you consecutively accumulate the gap score. So it will be -4, -2, -4, -6, -8. Then again on the column also, because again horizontal jumps or vertical jumps, both of these will count as gaps. So you go -2, you go -4, you go -6, you go -8. Okay. So that's good. That's filling out the first row and the first column according to the given values. But then what is the value for all of the other cells that are still present in this particular matrix, right? So the next uh, calculation you can do is for the immediately next matrix immediately next to this D here. Okay. So because for calculating the score for a particular cell, you need to consider three different routes. Okay. Now, what are the three different routes we'll just talk about? So the first row and columns are there, all indel or gaps. So you can fill out those. Starting for the top left cell, you can accumulate the gap penalty for each horizontal or vertical jump. And then, as I talked about here, you need to calculate three different routes to derive the scores for a particular cell. Okay. So let's say this four cell matrix is here. You are starting from this particular position, and you want to calculate the score for this particular position. Okay. Here, what you need to consider is you can get this score from three different routes. Okay. So what three different routes? So these three different routes, either you can go from I-1, either you can go from so I-1, J-1, either you can go from I-1 and J, this is the vertical jump, or you can go from I, J-1, which is this horizontal jump, right? Okay. So you start from this particular cell, S of I-1, J-1, and you calculate the score of Sij. You do the same for the other two routes, and whichever score is the best score, the maximum score, that will give you the score for this particular cell from the Needleman-Wunsch algorithm. And of course, as I talked about earlier, while you consider the diagonal sort of transformation from one cell to another cell, we will consider a match or mismatch score. For the vertical and horizontal jumps, you will consider a gap penalty. Okay. So in this particular case, what would be the value for this cell, let's say, right? So of course, you have to consider three different routes. That's one way of getting the value. That's another way of getting the value. That's another way of getting the value. Okay. So for this particular cell, again, so you start with a value of 0. Then you see, okay, what is there? So it's a D versus D. So that's a match, right? So match score is 1. So that means for this particular cell, the score would be 0 plus 1 equals to 1, right? Then if you see from the vertical jump, so that's this one. So it already had a score of -2 on the above cell, right? If you are going downwards, so you are again adding a indel or gap penalty, so that will accumulate to keep the total score of -4, right? Again, then this horizontal jump, again, what it was that starting from -2, and then again, since you are moving horizontally, you add a gap penalty. So -2 equals to -4. Okay. So only for the diagonals you are looking at a match or mismatch. If there is anything other than the diagonal, you don't need to look at the matching. You just go for a gap penalty. That's as simple. Okay. So here you have three routes. Of course, you start with 0. Okay. So I'll just so in this particular case, of course, you see that the score of 1 was the best here. I'll just erase this. And I will look at it from fresh. So what it was is that for 3 different routes, you first fill out all of the rows and then you get a score of 1, as I talked about earlier. So from the max scoring value, you got a score of 1, right? So that's the best score. The others were in negative values. Okay, so that's the best score. You write down that, you fill out this particular cell. And since now you have this particular cell filled out, you can also calculate the cell right next to it, because again, you need to consider 3 routes in all of these cases, right? 3 routes. So that's how you can get a score for this one by comparing all of these 3 routes. Okay, that's basically the same. You will get a score of -1 here, so on and so forth, so on and so forth, and you fill out all of the um, cells, right, in this particular case. Now, this is the Needleman-Wunsch algorithm, of course. Can anybody tell me what is the net alignment score for this one in this case? So we filled out the matrix using some given matches, mismatches, and gap scores. So what's the net alignment score here for the Needleman-Wunsch?
Sir, 5? No, or
No,
6.
no.
Mm.
So for the Needleman-Wunsch, the last cell of the matrix, right? The basically the rightmost and the downwards most, right? So the downwards right cell. So the last cell of the matrix that you are filling out, so that will represent the net alignment score for this particular matrix. Okay. So in this case, the alignment score is 2 here. Okay. That's how you get the Needleman-Wunsch score, the net score. Okay. Another thing that you have to kind of look for or keep track of is that it's fine, you get the optimum alignment score of 2, but how do you get the particular alignment? Okay. So for that, what you have to do is that you have to backtrack. So whenever you are getting a score from a particular cell, so you just keep an arrow on that side that you are getting the score for this particular cell from this particular cell earlier. Okay. So if it's a diagonal, then you just give a diagonal arrow. If it's an upward or if it's a sidewards horizontal value, you give a like corresponding arrow there. No? So that is basically to keep track of the score that you are getting. Now, how you calculate the or see the particular alignment is that you have to go for tracking the arrows throughout this particular matrix, starting from the your net alignment score cell. Okay, so let's say here you had this particular arrow that's tracking there. Let's say you have this one, right? And then you have a long stretch of kind of arrow in this particular range, right? Then you're going upwards, then you are going this way, so on and so forth. So that is kind of your if you track your arrows, you are getting something like this. Okay. So if you try to align it, let's say, so CATG, yeah. So CACT, so we are talking about this first one. Okay. So basically, you do it kind of in this way. So you go for writing your first sequence, which is, I think I considered this left one here. So CACT, okay. So the CACT part, you will see that this particular part is the same or not the same. So you had a CA here, okay. So that's from the diagonal here, right? So this diagonal, you are getting exact same match. So that is the exact same match. It could be mismatch also from diagonal, but that's fine. So the CA, you are getting the same here. So that is the match in this particular D region that is representative of this diagonal here. Then here you see a skip or a gap present here, right? So here, while on the particular sequence number 1, you have a C, you do not have an exact match or an exact character to put on this particular case for the sequence number 2 here. Okay, so that's for C versus gap region here, right? And then the next, again, you go forward with TGT here on the first sequence. So this TGT here again, and you see that this TGT is aligning with this particular TGT. See that there is a gap here. So the TGT and TGT could align together. And then again, you see a horizontal jump here. So that means that in this particular position for sequence 1, so this after this GT, uh, you have a gap here where on the other end of the sequence or sequence 2 you have a t inserted here, so it's a gap versus t here, and then the last element is alignment versus alignment, very same, right? So that's how you kind of get the alignment. You start to track the arrows from the very last cell. You start to see wherever there is diagonal you put the characters, wherever there is a horizontal or vertical jump you uh, basically put the gaps there that will represent the insertions or deletions depending on from which side you look, right? That will give the alignment, right? Uh, so anybody noticing something else in this particular plot? Something interesting perhaps that you could consider, yeah, for this particular alignment, of course. So if you look very closely here, you will see that okay, this position is fine, but from going from this particular cell to this particular cell, of course, we earlier went via this route. You notice that there was also arrows pointing from this cell to here also. Okay, so instead of going in that route, we could have also gone in this particular order, and then we would have met at the same point here and then went forward like the previous one. Okay, so that means that there was another possible alignment that we could have considered in this particular case, which would have looked something like this. Okay, so you see that the gap is present at different positions than the earlier alignment, but in both of these cases, you are getting an optimum alignment score of 2. So the alignment score is kind of the same. So that means that from let's say this coding system, you could get multiple alignments that have the optimum alignment score or value. Okay, so for that again, you need to consider whichever scoring, whichever alignment is kind of more preferable. Maybe you could have like different match score, mismatch scores, and so on. But again, just keep in mind that for certain values that you have set, you could get multiple alignments again that have the same optimum alignment scores. Okay. So in this case, these two routes we could have considered to get the same particular alignment. Okay, so I think we will talk about the next set of questions and so on in the next session. Okay, so yeah, so I guess today I will wrap up the class, and thank you all for joining. And then
Thank you, sir.
yeah, thank you so much. And then we'll talk about the next set of questions and concepts in the next next week's class. Okay. So again, as I said, that please try to look carefully, try to listen carefully to Professor Gomiha's lectures, all of the lecture series videos that is uploaded there on the NPTEL platform. And also there is another TA session that's happening for each week, which is scheduled on every Tuesday. So also try to tune into that TA session as well, whatever timing you find convenient based on your convenience, right? And then we'll also upload the TA sessions recorded videos to YouTube, and we will share the slides with the in the dedicated Google Drive folder, which we'll share with you via NPTEL. Okay. So yeah, thank you all for joining this week's TA session, and we'll look forward to the next week's classes, right, for week 4. Okay. Thank you so much.
Thank you, sir.
Yeah, yeah. Thank you all.